afternoon, dear colleagues. I want to present you some results of our investigation during 10 years. Water as in horizon of helioprotective means. I present now our maybe not so unknown institute, International Scientific Research Institute of Cosmic Anthropoecology. This institute was created 20 years ago on the basis Academy of Medical Science in Novosibirsk. Now our institute have a name by Academician Kaznacheev, because unfortunately 1 year ago Academician go out from life horizons of our Earth. So I will say you about invisible interaction between cosmic environment across protons, electrons, and other particles with living matter of not only of our planet, but planet or a sun system, maybe galaxy and universe. You know, yes, of course, what magnetosphere is a very important part for saving of our planet from direct influence, impact of different energies, protons and electrons from sun and galaxy. But during some more years, security function of our magnetosphere decrease. Every year, geomagnetic induction increased approximately 70 nanotesla. Our opinion is a very dangerous situation for our planet, for living matter, especially for our biosphere. And I want to show some result of our colleague about dangerous situation. You can see bed offshore, and you can look how change induction of magnetic field of our planet during some thousands years. Now we are here another cycle of geomagnetic deprivation now. You see how it depends level of geomagnetic induction with impact of different bioactive factors, for example, of chlor-36. You see, there is a mirror's case. In period of decreasing, magnetic field, we observe increasing of chlor-36. Also one sample, please. You can see how a period of decreasing of geomagnetic induction increase levels, streams, intensity of streams, different particles. Across data of our Siberian geophysics, Kuznetsova and Kuznetsov, we calculated data about different period of geomagnetic deprivation during some millions years. We can see how these different deprivation periods was according different genetic mutation. And for example, it is very completely to say in short time, for example, how appeared Neanderthals man, or disappeared Neanderthals and appeared, for example, Homo erectus. Before this, after this, you can say about new methods for study with influence of cosmic factors on the period of very sensitive to heliogeophysic factors, period of prenatal development of human organism, we created a special program with name Helios program, and calculated based about different cosmic factors, so as sun activity, number of valves, interplanetary magnetic field, geomagnetic induction, etc., in different period of prenatal development of human organism. And if we observe, for example, in our scale 7, it means what period of development different functional system of human organism was imprinted with very bioactive factors. For aim of creation preventive means a period of geomagnetic deprivation, we created special device, unique device for us. It is so geomagnetic installation or geomagnetic device. When we put our volunteers inside this device, we modulated situation with decreasing geomagnetic induction approximately 500 times. It is modulated situation with geomagnetic deprivation in future for our planet. And you can see what is the level of geomagnetic deprivation inside this device. After calculation of result of this experimental work, we found that different genes of human organism, for example, gene D4, you know, controlled activity of neurophysiological reaction in our brain. In this period of deprivation, appeared a new level of control of across this gene, different function of human organism, electroactivity of our brain, blood pressure, level of hormones, etc. We say maybe about phenomenon of geophysical expression of genes. Now also one result. After deprivation, we observe how different system, for us very interesting, creative work, memory, going in correlative depends from different spectrum of suns and galaxy particles, protons, different energies, etc. Its dependence appeared only at period experimental deprivation. When we investigate very important marker of biological aging, balance between carbon 14 and carbon 12, it is marker of biological age of our organism. You know that period with increasing aging, level of carbon 13 decreased. This is age, this is decreasing of carbon 13. But in conditions of deprivation, we observe two dynamic. At experimental group, it is decreasing of level carbon 13. At control group, we observe decreasing of carbon 14. Not decreasing, but it is maybe stable level. So, after this, we decided we must find very effectively mean for protection of heliomagnetotropic reaction of human organism, especially in this period of geomagnetic deprivation. And how I said, during 10 years, with different models, we test different variants of our experimental water. Water with helioprotective quality is our aims during some years. We use different methods for confirmation helioprotective effectivity of water. You can observe some methods is our original methods patented in Russia, and very many another methods. We created a special device, no very big device, so how I said, hypomagnetic chamber for people. It is a portable device with decreasing level of geomagnetic induction approximately at 100 times. When we put a sample of water inside this device, during some hours we observe what change, we must observe what change with our water. I want to present to you some result of this part of our investigation. You know, across very interesting presentation, Professor Korotkov, we used his method for control different, how change frequency, a period of very important for us event, solar eclipse, because we believe that period of eclipse this very interesting natural model for observation for galactic protons. Because you know what in this period sun part of proton from sun reflected of body of moon, but another part, approximately from galaxy, go to our planet. And for us it was very interesting observe biotropic effect of this part from galaxy protons. Here you see how changed during sun eclipse levels of frequency of sample of water. Why, but with using method of Professor Korotkov, we opened very interesting result when we study levels of his parameters for sample for water and correlation between protons in different period, not only in moment observation here, but some days or some months before moment of registration. And what is very especially very interesting for us, we opened significant correlation level significances here before registration for some days and some months before registration of sample of water. What is it? It is registration according to very interesting physiological scientist, Professor Anokhin, about adaptive strategy of biosystem, water to changing of cosmic situation, cosmic environment. But our opinion is only a hypothesis. It is if we can take a point of observation according to hypothesis Nikolai Kozyrev, Russian astrophysicist. He showed what what present and future time exist in, past and future time exist in present. This is a very interesting moment, maybe we can discuss this after in course. I continue my presentation. We used different methods for registration. What changed with our water when we put water inside this hyper-geomagnetic device? We registered difference between control water and water after our exposition with using polarization light. Also one method that we use for registration difference is method high effectivity chromatography. With using this method, it was registered our colleague in Moscow. We shown difference between control, this blue, and experimental samples of water after our exposition on the level of longevity cluster structure, what is shown across the method of Professor Stepanov in Moscow. Also, we registered increasing level of peroxide anion radicals in samples, control and experimental samples of water. And after this, we decided to do some experiment, how I said, with plants. You can see difference intensity of growth, our experimental and control plants during using different variants of water. Now, very interesting step of our investigation when we used our water for animals with rats, lamb, bister. These animals received water, two groups, experimental and control groups. We registered different parameters of these animals before, during and after experiment. We registered what after using water, after using also one very interesting tool, hologram. It is also an interesting method. Hologram with information about testosterone hormone. When we used this hologram for water, we registered after drinking with water, after drinking with water our animals, we registered significant difference between level of testosterone in different groups of our animals. Significant difference. How I said earlier, we registered in this experiment two level of stable isotope carbon-13. You can observe difference between control level of carbon-13 and experimental level of carbon-13 at animals that drink our water. This for us is a great surprise for us. We show how influence our water on the biothermodynamic process in living matter. And living system return to synthesis of carbon-13. After this experiment, we attempt to understand what increased level of carbon-14. And we show what for experimental group increase positive correlation between level carbon-13 and level of protons different energies, especially with energy 100 MeV. For us, it is a small discovery how change biotropic dynamic our animals under influence of proton streams. Here we show difference between different samples of cell culture of human. You see difference of density of and number of mitosis of this cell culture after using our experimental water, control and test. And also we registered what increased anabolic process in this system of culture, cell culture. And at period of sun eclipse at Novosibirsk, it was August 1st, 2008, our health volunteers include in our experiments. We registered different parameters, but for us was very interesting to observe how change dynamic, correlative dependence between hemodynamical parameters, for example, diastolic pressure, and different intensity of protons streams at period of our observation. It is period of, how I said, sun eclipse again. Before using of our water, we registered significant positive correlation between level of diastolic pressure and intensity of protons with different energy. But when our volunteers use our water, picture change. You observe significant but negative correlation between a level of diastolic pressure and the intensity of protons with different energy. Our opinion is demonstrated helioprotective effect of our water with using using this water. During some years we work here in Bulgaria. We attempt create here a center and during 3 years we worked together our colleagues from Varna with using different variants of our helioprotective water. Here we demonstrated how change dependence between parameters of blood before, during and after using helioprotective water. We demonstrated how change correlation between, very important for us, Röntgen influence of sun after using helioprotective water. Before this was positive significant correlation. After using of our water, we can look, we demonstrated significant negative correlation between parameters of blood, our volunteers, and levels of protons and electrons, and intensity of Röntgen influence of sun. We demonstrated these methods in other countries, in Germany too, for example, and we received confirmation with using different methods about effectivity of this system, helioprotective, and how for conclusion, I would say what during some years we present with methods across conference omics group in the United States on the conference Science of F and Climate Change. It was in San Francisco, Las Vegas, and now in this June in Spain, Alicante. And we propose this system, helioprotective water, to use especially in this period of cosmoplanetary changes. How I said in the beginning, the period of decreasing magnetosphere induction. It is very dangerous for our biosphere, and we must attempt to find solution together. Thank you very much for your attention. I am ready to answer, maybe you will follow up.
Time for some questions.
Yes, the details about your experiment during the eclipse at Novosibirsk is fascinating. Did you take the blood pressure measurements and things like that, let's say an hour after sunset on the same day, to see if there was any correlation between just the lack of sunlight and the cosmic ray and other influences from the rest of the universe?
Yes, thank you. Yes, we observe levels of geomagnetic parameters, for example, blood pressure, during, so it's a monitoring system, before 1 hour before eclipse, during different phases of eclipse, and we observe how increase level of the stellar pressure when begin main phase of eclipse. I demonstrated this in my slide, but after using of our water, dependence, not only dependence, but means of the stellar pressure and correlative dependence changed how I observe. It is a special method of geoecological monitoring. We used approximately 1,000 observations in different periods of sun and moon eclipse. Last moon eclipse was, September 28. We continue to observe and received new results.
Any further questions?
Do you find any moon phase influence on water quality?
Yes, it is very interesting. For us, moon influence, it is more unknown but influence of sun. We observe moon as reflected, how I said, period of sun eclipse, reflected streams of different protons from sun. At period of moon eclipse, we observe inversion of correlation between different parameters of human organism and level of light of moon. Maybe what for intensity of light of moon observe Nikolai Kozyrev. This is special work, how I said, and we find correlation in period of moon eclipse.
Right, thank you very much, thank you.